Yeah, I've done the same slides, like my what happened when I had my kids, and then yes, I will, and then I'm going to zoom through it, and then
Hello everyone, thank you all for coming. So today we have Professor Pam Pollock come to talk to you about her work with FGFR2 and endometrial cancer. So she does a lot of tests where she's treating things like patient-derived organoids with drugs and looking to see what happens as a consequence. The drugs she obviously is interested in are drugs that target FGFR2 and that sort of signalling axis. So I have known Pam for...
we went to uni together.
we
didn't really know each other then.
Well, yeah. We were in the same 30-year class as Brandon Wainwright. So molecular cloning, which is exactly what you're doing, we learned as well.
And we were in different pickup basketball teams.
Yeah, except, she played state, I played rugby. Anyways, I'll leave it up to Pam. She'll do a bit of an intro about herself and then she'll go into her science. So
okay, I just want to hands, I want you to put your hands up if you're thinking about the possibility of doing honours. Okay. And which one of those people, which of you is sure you want to do honours? Okay, good, that's okay. I'm the honours coordinator if you want to come up and I'm not the honours coordinator. Natalie Pachiniak is, but I am the faculty doing the grant proposal and the final thesis. I'm on the team, so if you want to come and chat to me at the end, you can. Okay, so as Sally introduced, I study FGFR2. I have got too many slides, so I'm going to go through it quite quickly. And but what I've tried to do is take like 1 or 2 slides out of a couple of different papers, sort of showing you the different sort of work we do. But I've tried to link it back to the cancer hallmarks, given that's your that's sort of the whole basis of this. So first of all, my career's trajectory. So I did my bachelor's with Sally at UQ. I then went, had a break actually before honours. I went to London for 18 months and worked on a genetics project. And then I did an honours project on cystic fibrosis, single gene, very, very easy. You know, I was looking at chloride channel functions in epithelia, right? And I thought cancer is way too hard. You know, single gene for me is what I thought. And then,
God, I thought that was me.
Sorry. Sorry, let me just turn that
off.
I can't even turn that off.
Press the button on the side.
I am. I'm not like... Okay. So then when I finished honours, I finished and I got a job in the way that science works, which is who you know. There was a postdoc in my lab whose partner was a QAMR and their lab was looking for an RA. So I went to a melanoma lab, even though I'd sworn I'd never work on cancer. I worked on melanoma as an RA for 2 years. In those days, you could backdate your PhD by a year, and I got a paper. And so then after, I ended up backdating my PhD, and I could include all the work in that paper as a chapter. I then met my husband, as he was a bioinformatician at work, on different floors, mind you, but I collaborated with him on a paper and then eventually went out with him. So it took a lot of convincing. So I got married. I then went to the NIH in Washington, DC and did a postdoctoral fellowship. I got a Australian C.A. Martin scholarship to go overseas and do that. And then rather than come home, because that's a 2-year there, 2 years home, I actually got recruited to a new, then I had my first child in my end of my postdoc. Then I got recruited to be a junior lab at a new startup institute in Phoenix, Arizona. A few years later, I had my second one. Then I discovered So what I was allowed to take with me from my postdoc to start my lab was actually FGFR2 mutations that I'd found in melanoma. And but they were loss of function and it wasn't working like dogma would suggest. It's a kinase, how can it be a tumour suppressor gene? So it took me years and years to try and work that out. And also I was changing from a really genetic background into now trying to do cell biology, even though I'd never myself run a Western blot, you know. So I was really lucky and managed to recruit an Australian, Rowan, from my PhD, who was in the USA, and he came in as my sort of senior lab tech that had done all the techniques I needed. Because we were studying FGFR2 and melanoma, we then looked at it in other cancers and found it in a couple of cell lines, endometrial cancer cell lines. And then I started a new collaboration with someone who had a large cohort of endometrial cancers with outcome data. So that's sort of how I got into endometrial cancer. And so then I kept doing that, had my third child, and then I came home in 2010. And, you know, I always thought I'd work on melanoma and endometrial cancer because that was my background, but there were so many other melanoma researchers here in Australia. It was really, really competitive and nobody wanted to work with me because they all had these big program grants that don't let you then be a CI on someone else's little project grant. And then the other thing was when I came home, there was not a single lab in Australia working on endometrial cancer. It affects grandmas, you know, and women, and is understudied. So I thought I could make a bigger contribution to science by studying endometrial cancer. So I was research only for a long time, for 10 years here, and then in 2020 I've converted to research and teaching.
The dark side.
The dark side, yes. Okay, so now here's the thing. Can I just go one forward? Yeah, you can ask me questions after this. Here's the thing. What people don't talk about is that I've had pretty crappy mental health at various stages during my career, and my first major episode of depression was in honours, and I ended up actually taking 2 months off and getting on medication and finished like 3 months out of sync with everybody, but at least I finished it. And then weirdly at the end of my PhD, after I'd written up and submitted, after my second kid, and actually the year I came home, it was hard to adjust to coming home. And then I probably had my worst one in a long time in 2023. And I guess I just wanted to put it out there that, you know, your prime, you know, this next 10 years is when, you know, you struggle with lots of things, or you may have already. And I just want to say that you can have a career in science, you know, and deal with some mental health struggles. We don't talk about it enough. It's, I'm sure I probably haven't been as productive if I hadn't had them, but I think it is, rather than any sort of job that's client focused, where you have to put on a happy face and, you know, if you're a GP or something, you know, and you just have to keep seeing people. You can have patches where you're a bit down and hide yourself in the office and just sort of, you know, do some thinking, or, you know, do research without having to really be out there and interacting with people. And I don't think I could have survived in very many other jobs, but I love this. So it's, you know, but this was just really to say, don't feel bad if you have ever struggled with your mental health, because, you know, I'm a big believer in pharmacology, but, you know, you can do anything. Okay, oncogenic hallmarks of cancer, right? You know this. So what I'm going to try and do is talk about the fact that I've said here EGFR and KRAS, and I'm trying to try and show you evidence that FGFR2 is also a major oncogenic driver. It functions differently in different tumour types, and therefore we haven't yet got an FGFR inhibitor approved in endometrial cancer, and I've been trying, but we will get there. So it's a cell surface receptor, much like EGFR, and it can be activated by lots of different ways, and there seems to be tissue specificness to the way that it's activated. So cholangiocarcinoma is tumour of the bile duct, weirdly, a bit close to the liver, and it mostly has translocations. that, put FGFR2, join it with something that dimerizes, and so then there's just these dimers formed all the time. Other things have amplification. What we have in endometrial cancer is point mutations that turn the receptor on, and actually hopefully I'll show you 1 or 2 slides of altered splicing. So this is, so I found FGFR2 mutations, as I said, in a couple of cell lines in a big screen, and this is the old days. We were doing this by Sanger sequencing. This was amplifying every exon by hand. So this was our first cohort. So I reached out to someone, didn't know him from Barra Soap. He's now become a mentor to me, and actually a good friend. We've been still now. What's that? 20 years, probably 15 years later. So he had a great cohort. We had hotspots, which is really good proof that it's an oncogene, because there's only specific ways it can get activated. When you have a tumour suppressor, it's sort of all over the place. And so we also screened for KRAS, and they were mutually exclusive, which pretty much told us that FGFR2 signaled through the MAP kinase pathway more than anything. And they seem to be more common in a subtype of endometrial cancers, which were MSI, so microsat instability, or MMRD, mismatch repair deficiency. So this, I'm not going to do this in the order that I did the studies, but so this was actually sort of probably about 9 years later, we finally managed to complete a multi-institutional cohort. Because if you're trying to get a biomarker approved or a theranostic, something that will indicate therapy choice, you have to do it in one single institutional cohort. Then you need to verify that there wasn't something weird about the samples from that place. So you have to do it in a multi-institutional cohort. And this was 20 gynae cancer centres across the US. I did this while I was in the US and finished it when we came home. And so again, you can see these hotspots. There's one, this S252, I'll talk about that later. It has, its effect mimics the splicing change that I'm going to talk about, which is why we've been looking for it. And these ones in the kinase domain obviously just turn the receptor on. Some of the ones in the transmembrane domain, they turn it on, they cause dimerization, and some of them mislocalize, but I took that slide out because I had too many. So then, because this was a good cohort that had outcome, we could say, if you have an FGFR2 mutation in univariate analysis, do you have an increased chance of your cancer coming back? Because the majority of women spot postmenopausally, and women are much better about going to the doctor than men, and so they get their endometrial cancer diagnosed early. Okay, it's symptomatic, unlike ovarian cancer, which is, everyone calls it the silent killer because there are no symptoms, whereas postmenopausal spotting is a primary symptom, not of everybody, but about 70% of people will present with that. So the hazard ratio is 2, so you're twice as likely to progress if you have an FGFR2 mutation versus those that don't. And then we did multivariate, which takes into account age and all the other things that make your cancer worse, like late diagnosis, deeper invasion. And even if we took all those other known things into account that were all we associate with your cancer coming back, FGFR was still significant, that it definitely drives more aggressive cancer. And so that was really important for us to be able to say, these are the cancers that come back, and so these we really want to try and get an investment into a clinical trial. So what I've tried to do is I've done some screen captures here of our hallmarks, okay, as I've gone through. So this is a standard IC50 curve, and at the top in the green and blue are wild type cell lines, and at the bottom are 3 of our cell lines that have FGFR2 mutations. And you can see that only they are sensitive, okay? And we sort of call this oncogene dependence. Now, how you measure this can be lots of different ways. Proliferation assays that measure ATP, that measure protein, that measure DNA content. This is actually a measurement of viability, so cell death, or actually alive cells. So we know we got an inhibition of proliferation when we inhibit this kinase, which is what everyone thinks of as kinases drive cell proliferation. But when we inhibit it, we also get cell death, which is great. So that told us that they were sort of addicted to this receptor, and in the absence, if you block the signalling, then they just fall over and die. And so that was exciting because we thought, well, that's going to be a more effective drug in patients if we're actually killing the tumour and not just stopping it growing. Because, you know, the patients want a cure, they just don't want to have their tumour there and not grow, obviously. So, and so then if we just go back here, the next few slides are on this resistant cell death. So one of the things we wanted to do is what do we combine an FGFR inhibitor with to make it even more effective? And I was all about the fact that we get this cell death. Can we make the cell death more? In order to do that, we need to know how the cell death is occurring. So this was a really detailed mechanistic study with lentiviral knockdowns of different genes and overexpression of different genes and all sorts of things trying to actually work out. And I'm only going to present a couple of slides. But one of the... Oh, yes, sorry, darling.
Can you check back to that last slide? Those 3 cell lines that you have, the FGFR2 mutations, are they 3 different types of mutations?
Yes.
So you're talking about like the transmembranes?
Yep, so one of them is transmembrane, one of them is kinase, and one of them, actually no, 2 are kinase mutations. And you had a question before.
My question was to do with like the research in endometriosis. Are there more labs now? Like how many?
So there are. So endometriosis is weirdly not related to endometrial cancer.
Yeah.
You know, it actually is related to ovarian cancer.
Yeah. Okay.
So there are more labs studying that now.
Yeah.
I think in my last 20 years of my career,
Yeah.
there's definitely understudied women's problems have been that men just went, you just got bad period pain and you're just a wuss and you can't cope. You know, suck it up, love. So, you know, one of my kids has got endometriosis, but, you know, you have to confirm it you have to confirm it with a laparoscopy,
yeah.
And you know, you don't necessarily want your young girl to undergo surgery. And so, you know, most gynaes now said, you know, yes, if you're on your floor in fetal position for 3 or 4 days, let's put a marina in and see if we can stop them, because that's just, we don't need to do surgery to confirm that, you know. So there's genetics going on. I did one thing, because there was a big GWAS study in endometriosis, and there was a peak, 10q26, right over FGFR2. So when I first found it, I was like, I knew someone who studied endometriosis, read his paper, and he'd worked with my husband years before, so I wrote to him and went, I think I've got the one that's under 10q26. You know, don't tell anyone, but I found mutations in this. Can we look for SNPs around FGFR2 in your GWAS thing? Big study, took like 9 months, freaking zero. Nothing. And I was like, how can that be? I was sure it was going to be a huge player, because the peak was right over it. Oh, you know, as far as you can, it was under the peak. But yeah, no, they're not really related. Yeah.
Does this type of cancer only affect people post-menopause, or can it affect others as
well? No, like most cancers, there is a familial genetic syndrome that gives you a risk of both endometrial cancer and colon cancer. And it's a mutation or aberration in one of 4 genes that are involved in mismatch repair, in the DNA repair. You'll have a seminar on that later. And it basically means that the proteins that are looking at the DNA as you replicate it, to look if you made any mistakes and then correct it, they're broken. So you just accumulate mistakes all the time, and therefore those women get cancer really early. So those women might have cancer in their 40s or 30s.
Is FGFR proliferation always indicative of cancer, or do you have to rely on other biomarkers as well?
So I think FGFR2 is an early driver, right? But alone it's not enough. And the reason is now they've done some really cool studies of spatial genetics in normal endometrium, and so of women, of women that are 20, 30, 40, 50, 60, 70, 80. And when you shed your endometrium and repopulate it with stem cells, you get areas of the endometrium that are all populated from a single stem cell. So when they did the spatial, they could see FGFR2 mutations in normal endometrium contributing to the glands in an area. And it's not in very many, like 10% of normal endometrium, no, probably 5% of normal endometriums, but it increased the older you got, so you had more chance for one of these stem cells to accumulate an FGFR. But because they were normal endometriums, obviously it's not enough alone. So you need other things to happen for the cancer to occur. Yeah.
But you're saying that it actually indicates what a more aggressive cancer as well as a cancer that would reoccur.
Yes. So we were looking at sort of early stage. So that means just in the top, just in the uterus, hasn't gone through to the muscles underneath the uterus and hasn't extended down into the cervix. So that's stage 1 and stage 2. Stage 3, of course, is sort of lymph nodes. Stage 4 is distant mass. So you had a higher, I'll get to you, you had a higher chance of having a mutation if you presented with metastases already.
Okay.
And
What
or
about?
if you,
Oh, sorry.
or if you actually had presented early and got a hysterectomy and you were cured, and then you recurred, that had a higher risk. You then was close to 20% chance
Oh,
you had
wow.
an FGFR2 mutation in
Okay.
the patients that recurred. That doesn't seem to be working out. So for enrolling into clinical trials, it's been hard to find FGFR2 mutant people. So what we think happens is that they do recur more, but often they might have regional radiation or chemotherapy, and we think that they actually respond a bit better to that because they're not in the they're not in the people that have recurred and they're not as common as 20% in the people enrolling into clinical trials.
Yeah.
Sorry, you said
I'm going to go to this one. You had your hand up in the
Yeah, I was just going to ask, so is genetic one of the biggest risk factors for endometrial cancer?
Do you know what the biggest risk factor is? Obesity. Huge obesity. There's different types of endometrial cancer, which is why I didn't talk about that. There's like 4 major types, and obesity is associated with the 2 that are most common and are responsible for like 80% of all endometrial cancers. So I don't know. I went to a fantastic conference last year in New Zealand on gynae cancer, and a lady got up there and said, what is the role of us in prevention? And she said, if you have a BMI of 42, which is pretty big, right? You're obese. So I think it's 25 to 30 is overweight, 30 to 35 is obese. I think then 35 to 40 is maybe morbid obesity. So 40 is you're pretty heavy. You have the same risk of endometrial cancer as a smoker who's smoked 2 packs a day for 20 years. So it is a really, really strong risk factor, and every 5 BMI units, your risk doubles. So, me at 30, I've got twice the risk. I was 35 once. Then I would have 4 times the risk, but I still have 2 times the risk. So there's been studies that if you got everybody to lose, 5 BMI points, like 10 kilos, 5 BMI points, you would halve endometrial cancer in the world. Like if you think about it, because it goes up every 5 BMI points. Yeah.
I was wondering, when you were talking about different types of endometrial cancer, is that based on the cell type which they're derived from?
We don't know. So we think the easy question is we don't know yet, right? There's been single cell RNA-seq studies now which have identified multiple types of epithelial cells in the endometrium. We haven't yet been able to link the genetics back to which stem cell type, There's going to be glandular, like they're basically at the bottom of the endometrium, and they're hard to identify. In some cancers, they're really easy to identify. It's very contentious what the markers are to identify the stem cells. But the major subtypes are mainly how they're driven, because MSI tumours have this high mutational burden by having the mismatch repair genes defective. There's a type called copy number high, which has such a low number of mutations, like maybe literally 5, genes that are mutated, and it's almost always p53, which obviously has lots of functions. But what it has is crazy chromosomal aberrations, right? And so it's got the worst prognosis. And then you have this one that's got a little bit of chromosomal, but no MSI, and it's called NSNP, so no specific molecular profile. And then you've got 10%, which is due to a POL-E mutation, which is one of the polymerases used to actually replicate your DNA in mitosis. And that has crazy high mutation rate, like literally every second gene has a mutation. And they have the best prognosis, which is counterintuitive, right? And so we think that they have so many tumour epitopes on their surface that the immune system can see them so much better, and or so many genes are broken that they actually fall over with chemotherapy, because they're so messed up. So, yeah.
Interesting. Sorry, I know you want to get back to this. In terms of the FGFR, like the inhibitors, have you looked at like FGFR like 1 or 3 or like any other numbers, or is it only
so we
2?
did. Other people have done it.
Yeah. Okay.
Yeah. So, but we have. I am not actually presenting it, but there was a research-only drug. This is one of my very first figures, and it was so beautiful. I keep putting it up. So this was a research drug. There were no inhibitors approved. Then there were dirty inhibitors approved, which inhibited then there were dirty. I went through a phase of studying dirty inhibitors with FGFR activity because that's all there were. Then there was about 5 companies that made pan-FGFR inhibitors that had very little activity against anything else, and they've gone in and got approved. So there's 3 of those approved now in other cancers. And now I'm actually moving, but they have quite a lot of toxicity due to hitting FGFR1 and FGFR4 gives you horrific diarrhea. So then people have tried to go, I'll just have 1 to 3 and not 4. But 1 has lots of hyperphosphatemia and you get calcification in your muscles, so they're trying to get rid of 1. So I've got working with a drug now. We haven't put it in a mouse yet, but it only inhibits FGFR2.
Okay.
So, and it's been done in cholangiocarcinoma, had exactly the same response rate as a pan inhibitor and a ridiculously reduced side effects, like hardly any treatment adverse above 3. So 3 and 4, you need dose reductions because you're so sick. 1 and 2, you get a bit of nausea, So they were mostly 1, 2, 3, 4s. Like from maybe 50% of people having adverse side effects that were 3, 4s to like less than 10. So I really think that my older ladies would appreciate the going on a specific FGFR, and also we want to do a combination. So of course, if we only do FGFR2, we'll be better able to combine it with something, and you'll have increased tox by combining it anyway. So you may as well get at least tox from the FGFR. Yeah, and then
And that'll be the last 2 because seriously,
this is like my second slide.
Yeah.
Okay, you go.
I was just going to ask, you said that there were the pan ones, but yours is specific to 2. What did you target to make it specific to?
So there are labs that you'll hear talks from
Yep.
that have tried to, and Derek's one of them, who has tried to make his lab do everything, and one of them is medicinal chemistry and designing their own drugs. I went, no, I am wanting to get treatments into patients as quick as possible. I want pharma to spend the 10 years developing the drug, and then I will test it in my models.
Yeah, that's fine.
So, yeah.
Sorry, I was just wondering about the link to endometrial cancer. So it's the FGFR2 mutation, but how come, is it just you're specifically looking at endometrial cancer?
So yes, so I looked at FGFR2 mutations in 1,000 cell lines, and endometrial cancer was the only cell line where I found a mutation rate of 30%. Literally, we had 10 on our panel of 1,000, and we got 3 out of 10. So I was like, ooh, that's cool. And they were activating, whereas, it's randomly mutated in cancer as passenger effect, and there are other cancers apart from melanoma that have loss of function mutations. But yeah, okay, can I keep going? Okay, I'm going to have to whip past this, right? So this was just basically, everybody was saying, lots of people were reporting this in many cancers because it's actually 7% of all cancers of all cancer types have an FGFR activation. So every cancer has a few with this. And people were saying it only inhibits proliferation, doesn't cause cell death. But they were using a caspase-3 kit, mostly, assessing death by caspase cleavage. We looked at that and we didn't see any caspase cleavage, but when we did an annexin 5 flow cytometry assay, so when cells are dying, they flip one of their membrane proteins to the outside and you got an antibody to it, and you can say which cell, you can detect the number of cells that are in the process of dying. And so we could see that, and a lot of people did caspase at 48 hours and said there was no cell death with FGFR inhibitor. Yeah. And we did annexin staining at 24, 48, 72 and 96. Nothing at 48, started coming up at 72, 30, 40% by 96. So it was much later than the EGFR, because EGFR is 24 hours. So basically we went through all this literature, and we were thinking a lot of people are missing it because they're just using a caspase kit. Okay, so we wanted to investigate it. So we got no caspase cleavage, even though we were getting cell death. And so that means that it's not classical apoptosis, right? Because classical apoptosis involves caspases. And so there was an inhibitor called ZVAD, which inhibits all caspases. No, inhibits all caspases. So we treated with ZVAD, and sorry, let me get my glasses on. So we treated with ZVAD, and down the bottom, ZVAD alone, got a little bit of death compared to DMSO. But if you compare those three, the middle one is now clinically approved, FDA approved. There's no reduction in the cell death. If I go here, there's no reduction in the cell death when we completely inhibit the caspases. So we know that it's a caspase-independent type of cell death. And so that means it's not classical apoptosis. And we got this in a pretty good journal. We did heaps of other experiments to show that it was mitochondrial dependent by overexpressing and knocking down BIM and all these other things. But, so that was a big thing that people had missed in the field. And so then the question that I got when I presented to seminars or whatever is, but do you know your cells are competent to perform classic apoptosis? Because there was a big furor. One of the breast cancer cell lines didn't.
MCF7.
Which one?
MCF7.
MCF7 is used, huge breast cancer research. It actually has a mutation, I think, in one of the caspases and doesn't undergo apoptosis. And people spent so much time and effort investigating this new cell death in MCF7s, and it was actually just that they were mutated in a critical protein. So everybody was saying to me, you have to prove they can undergo classical apoptosis, and then we'll believe that FGFR inhibitor doesn't need them. And it's not an artifact of these actually being impaired in apoptosis generally. So it took actually quite a lot of inhibitors to try and get one that killed well and via caspase. So each of our two cell lines, we had to get a different drug. One was Starsporin, one was actinomycin D, and you can see even 8 hours it's inducing caspase cleavage, and you can block it if you're down the bottom. This is annexin positivity, so our assay, there's our assay on the bottom and caspase cleavage at the top. And so you can block the death from actinomycin D with ZVAB. So we know they can undergo classical apoptosis, and our results were not an artifact, and we absolutely had to do that. And that's the sort of detailed mechanistic work that's part of cell biology. And if you design it well, you get really clear answers that, yes, this is what's the truth and not. And so this second cell line, actinomycin D didn't kill it, so we had to use something else. And you can see in the bottom, the diasporin absolutely induces death at, really high death, 40%, at like 8 hours. So, we were looking at 72, 96, really strong inducer cell death, completely block up with the caspase inhibitor. So that was crucial. And then actually there was a drug that is involved in apoptosis, ABT 737, it's approved. We were saying once we knew it was mitochondrial dependent, this is a BCL2, BCL-XL inhibitor. We're like, will it be more, now that we know it's caspase independent but mitochondrial dependent, can we use this inhibitor that affects the mitochondrial BH2 proteins and get better cell death? So we did. The first bit of the column are three different FGFR inhibitors, and then the Abbott drug by itself. And then when we combine it with this BH2, we got much more cell death. And then this is in vivo. So this dots over here, this is actually cleaved caspase. And so you can see by itself, this is 4 days of treatment, and the mouse was sacrificed 4 hours after the last treatment on day 4. And there's just very sparse cell death. So there's always cells turning over anytime. So the FGFR inhibitor, the second one, and then that Abbott, the mitochondrial BH2 inhibitor, and then with the combination, it's synergistic. We get a much better cell death. But these two drugs are made by different companies, and they weren't really interested in doing a clinical trial, which is unfortunate. So there was just another one. Again, this is an in vitro colony assay. Gave us really beautiful results. The top is 3 different sorts of PI3 kinase inhibitors, which is a major signalling pathway downstream of receptor tyrosine kinases. And a lot of endometrial cancers, about 80%, have a PTEN loss or PIK3CA mutations. So we knew that they had FGFR turned on, which was activating the MAP kinase pathway, and also PIK3CA pathway was on due to PTEN and PIK3CA. The bottom one here, this first one, is FGFR inhibitor. Now, I am a strong advocate. If you ever do honours, I will ask you a question about this. What is the clinical dose that is approved in the clinic? Because you should be doing your in vitro experiments at that dose or at that plasma concentration that patients get. Because anything can kill if you put 10 micromolars in or millimolars in. And
Including water.
Just saying. It pisses me that there are still papers that I review that use, like an absolutely non-physiological amount of drug and then go, oh, I found a cancer cure. So I actually always go and find the phase 1 clinical trial, which is why I like using pharma drugs, especially once they've passed phase 1 and that data's published. And so the plasma Cmax at day 15, which is after 2 weeks of drug, because you sometimes get a slow accumulation of drug, is 450, and so we use 300, right? Also, we were trying to do a combination. We wanted to reduce toxicity, so we wanted to use the least amount possible because we wanted to use it in combination. And weirdly, the GDC PI3 kinase in people, in their plasma, 2 millimolar. Hugely more, like I would think unphysiological, but it dissolves in the plasma really well. It's not super bound up by binding to other proteins in the blood. So, and again, but these have terrible toxicity in patients, right? And they cause diabetes because all the insulin pathways involved is blockade. So I didn't want to be treating my patients with 2 millimolar. I wanted to see how much can we reduce that by if it's synergistic with FGFR, and will that work? So no real. So this is really an assay. We treat with drug, and then after 72 or 96 hours, we then count them so every plate's equal, and then put them on this colony or a plate. So the PI3 kinases at these low doses does nothing, but if you combine that with a low dose of PGJ, you get a huge increase in cell death. And so that means that there weren't any cells that survived to form a colony. So it's a really good survival assay because you basically plate down the cells that are left at the end of your drug treatment and put them in tissue culture, walk away, feed them every 3 days and come back 2 weeks later, do this little quick crystal violet stain, and that'll tell you how many cells survived your drug treatment. And so this was super cool. Again, so there was a clinical trial because I did these because they're all made by the same company, right? Clever, same company this time. So there was a clinical trial of the middle one, BGJ and BYL06, because this is specific to one of the PIK3CA pathways, so it would be theoretically not as toxic as pan-PI3 kinase, but together horrifically toxic in patients. So they had to just reduce, and they basically stopped the trial because they couldn't get, a dose. I don't know whether they were using a teeny tiny amount like I was in this assay, or they were starting with what was approved by itself. But basically, this wasn't. The PI3 kinases just haven't really panned out very well in most cancers because they're just so toxic. Okay, quick. So oral Gavara. So this is in vivo. I really loved moving into in vivo because things work in cell lines and in culture, but you have to prove that it works in an actual living animal, right? It goes through the vasculature. It doesn't get metabolized and go away. So this is vehicle in pink, BGJ in orange, and the combination, for the first time, we actually, almost all papers reporting in vivo results show a slowing in tumour growth. But in a clinical trial, that would be progressive disease because their disease didn't go away. It kept growing, okay? So if you can imagine a straight line across from one, I call that the line, right? So you have to get a drug combination that goes below the line because that's the tumour shrinking in vivo. And that's the tumour actually getting cured. So lots of things that are aimed at just inhibiting proliferation can slow it down, but they won't make it regress. And that's why I was really concentrated on its role in cell death rather than just proliferation. Okay, I'll totally skip through these next two ones. It's also involved in invasion. For this, we had a cell line, no FGFR mutation, super well differentiated. It's like a really early cancer. And then we put wild type in and a couple of mutants to see what it did. So this is it's a bit like NCF7. It's a very benign cancer cell line. It's not very progressed. And so what I'm getting you to look here is the pink in the middle column. The pink is Golgi. And so these cells have beautiful basal apical alignment. So their bottom is in the middle. No, their bottom is on the outside of this lumen because it's a gland we're mimicking. The inside is where you would secrete, or basically the inside of the uterus. And so the Golgi are lined up. And then on the outside, on the last column here, this integrin, this pink stain is on the outside. And that's the basement membrane. So they can form beautifully polarized. So if you've studied EMT, epithelial to mesenchymal transition, which is later in your things, it's called phenotypic plasticity, it is actually one of the things you have to get to invade in a cancer because normal epithelial cells are polarized. They have a direction, they sit up, they have a bottom and a top. To become a cancer that's capable of invading through the basement membrane and going into the blood, the vasculature, you have to lose your top and bottom, and you don't know where you are, okay? And so this was wild type. The bottom one is wild type. The top one is nothing. So by putting in FGFR2, it looked pretty much similar to empty vector. Now look what happens when we put, so that's the bottom one is also on this page. These are the two, that's the most common kinase mutation. And basically it was just invading into the 3D matrix. So it was more invasive, but very coolly, if you look in the middle column, its Golgi were completely disorganized. It had lost the top and the bottom of where it was in the world. And so that explained why it was super invasive. And the bottom one is a different mutation, and its Golgi were even more stuffed up. Totally didn't migrate. And one more thing, and I'll get to you. The thing that was interesting, as these two different mutations actually invaded very differently. Okay? If you look on the last column, the wild type has the lovely pink basement membrane on the outside. Neither of the two mutants have a basement membrane, have any polarity. They don't have a bottom. They're totally messed up. But the middle one actually sort of shot out little like invasive little arms. And the bottom one, which is a different mutation, never did that and just sort of grew bigger as like a blob. And so I would think, and we haven't got enough patients with each mutation, but I bet that middle one would have worse prognosis than the bottom one if you had 100 patients with one mutation versus the other, because that middle one has got that really invasive behaviour in vitro. And yeah.
Was that Ishikawa with the mutation of FGFR?
Yeah.
Or that just Ishikawa?
No, that's Ishikawa, where we put in you're doing cloning.
Yes.
We cloned wild type into a lentivirus so we could transduce the cell line, not transfect. We transduced it with virus because that gets you better transduction efficiency. Then we made each mutant, we cloned, so we had actually wild type. Then we did site-directed mutagenesis to introduce each mutation specifically. And I've got a bunch more mutations and I've got no stuff to do. So the next thing is super cool. And how am I going to time? Crap. So this is probably not unique to endometrial cancer because other people have reported it, but we've really shown that it's associated with poor prognosis. So this receptor, and I don't have a better picture, but basically on the outside bit, that's talking to the ligands. There's two exons that make up part of where the ligand binds. One of them's encoded by exon 8, one of them's encoded by exon 9, and there is mutually exclusive splicing, 7, 8, 10, or 7, 9, 10, and that is controlled exquisitely in a tissue-specific pattern. So epithelial cells express 8, and have that FGFR2b form. Mesenchymal cells, the underlying stroma or that fibroblast in the muscle, express FG9, transcribing 9, and express R2C. And the way that works is it's very exquisitely paracrine communication. So the epithelial cells expressing B make the ligands for the mesenchyme that don't transactivate themselves. And the mesenchymal cells that are expressing C don't make any C ligands and make the ligands that transactivate B. That S250 252 mutation I talked about in the middle that had that super high SP, that mimics, it's actually in accurate people. It had been studied before I even got to this field because it actually occurs in patients with the worst developmental syndromes. And that mimics this. So in people, they've shown that if you have that mutation, your C could bind B ligands and your B could bind C ligands. So based on that, we had a hypothesis that if that mutation is common, surely, get that, incorrect splicing in the epithelial cancer cells would mimic it. So they should only express exon 8. If they've switched and now express exon 9, then they will be able to turn that receptor on because they make the ligands for it. and lead to constitutive receptor activation. But instead of a kinase mutation which changes the conformation so it's always on, this is a splicing change, which means that it's always on because it's making the ligand. Yes?
Do you know if it's the same splicing factor that gets you the differential, like 8
No.
versus 9?
So no, they're different splicing factors. Basically, exon 8 has a really crappy exon acceptors. The beginning of exon 8 is not a very good splice acceptor. It's not consensus, right? Because there's a consensus sequence. The one on exon 9 is a consensus sequence. So in order to express 8 in epithelial cells, there's a protein called ESRP1 that binds and basically covers the exon splice starting at exon 9. So as the splicing machinery is going on, it can't see exon 9, so it goes to exon 8 and then joins exon 10. And what we've found is that some of our endometrial cells have loss of function of ESRP1. So yes, I want to keep going.
No, that's because the isoform's switching. Do you have to target both at the same time then?
No, so we think, well, there aren't drugs that target one or the other because this is extracellular and the drugs inhibit the kinase domain. Theoretically, if we had a monoclonal antibody that only bound the C form, we could inhibit one and not the other, but that would inhibit FGFR2C everywhere else it was expressed as well, not just the cancer. But anyway, so we have this, so my postdoc, did optimise this brand new assay, which this wasn't available 10 years ago when we found the mutations and I initially tried to start it. We were doing RT-PCR and it didn't work. But this little red dot says a cell expresses the C isoform. And it's basically a 20-mer, an oligo, that's 20-mer, that has 10 on exon 7 and 10 base pairs on exon 9. And so it is at the exon junction, and the only places it'll bind is cells that have an RNA that joins 7 to 9. And so then we did that, and then basically we did this on a huge cohort of tumours that had all their outcome in all molecular subtypes, and we could show that these are the 4 subtypes I talked about briefly. So the red one is the one with the chromosomal craziness. The blue one at the top is the ones with every second or third gene mutated. The orange one is the ones with this MSI. So they have a pretty high mutation rate, just not crazy high. And within that group, if you have this alternate splicing of FGFR2, you are way going to, so the dark blue line here, this is the people that are progressing. So you are going to progress quicker and actually die quicker. So this is disease-specific survival. So every little notch down is someone dying. Okay? So if you have this spliceoform, you're much more likely to die of endometrial cancer than if you don't have it. Again, hopefully a bit of evidence to convince a pharma company that it's worth freaking trialling an FGFR2 inhibitor in this patient population. Anyway, then we made PDXs, so that all that work was done in cell lines up until that splicing stuff that was done in tumours. So the last probably 7 years, because I'm cherry picking, I've really been working on trying to do this in patient-derived xenografts, which are more reliable and more reflect patient tumours than cell lines that have been cultured for 20 years, and then patient-derived organoids. So quick. So this is PDX models with the splicing, because once you grow it in culture, you mess with the splicing anyway. So we needed to do it in PDXs, always grown in mouse, never seen a culture dish. And we screened all of our PDXs, found the ones that had splicing. And we only published this last year, or 2023 maybe, if you have the splicing switch, you are also susceptible to FGFR inhibition. And the good thing for us is the mutations maybe happen in 10-20%, depending. But this splicing switch happens in more like 30 or 40%. So it's a larger patient population that might benefit from FGFR inhibition. And then this is actually introducing another hallmark, which is activated FGFR, and this is, the last one is mutated, the first three are spliced. The top, that brown staining, is immunosuppressive macrophages that turn the immune system off and basically stop the CD8 T cells, effector T cells from killing the cancer. And if the bottom one is FGFR inhibitor for 7 days. So you can totally change the immune landscape so that the immune system can see the cancer. So what I'm doing right now is I've got humanised mice, so they get irradiated as if you were getting a leukemia transplant when they're 2 to 5 days old, which obliterates the mouse immune system. Then a very special technician at Tri injects 50,000 baby hematopoietic stem cells obtained from baby cord bloods where they didn't get enough to go into the cord blood bank to treat actual patients with leukemia. And so there wasn't enough volume to treat it, so they're sort of discarded as excess, or they're eligible for us to do it. And only not every model has the most common haplotype, because you've got to have the partial matching of the haplotypes. So then she injects it into the portal vein of the liver with a baby pup. I don't know if you see the mouse pup, it's about that big. And she just like knows where to aim it. And she puts that human baby stem cells into the mouse, then doesn't do anything for 8 weeks, and then she checks, she basically does a retroorbital bleed and runs it on a flow machine and says what percentage of its immune system is human versus mouse. And so these have got between 25 and 75% human immune systems. And now we're going to try an FGFR inhibitor and the approved immunotherapy. And they should show way better activity in FGFR2 mutant models or FGFR2 spliced models than the immunotherapy alone. And so, but a lot of them have been dying of graft versus host disease. And so that it's not a great, it's donor specific. So we got one baby cord blood, we got 13 mice out of it, and they all survived. And the current one, we're on cohort 2, we got 10 mice, which was how many? Well, she maybe did 12. A couple died before they got to 9 weeks when I get them. And then now 13 weeks. I haven't been able to do the experiment, and we're down to 2 left. But so it's just been a weird, for some reason, that person's immune cells, that baby's cord blood, was very attacky of the mouse immune system, and it's just model specific. And she said she's never seen one this bad, ever. So I've got some more coming in October. And then, we'll know by February.
And unfortunately, we're going to have to wrap it up there.
But I can answer any questions outside. Sorry.
As usual.
Well, I think we should thank Pam before you go.
Okay.